Hi there and welcome to lecture 9. So this is our last lecture and we're going to be finishing up the linear model essentially by looking at the anova function, which is a way to test whether factor variables are important when we have a linear model. So you remember from lecture 8 we were looking at linear models with factors, and we looked at the example of the Petrels dataset where we had the 6 different areas. Where from our box plot here we can determine that there does seem to be some difference in the average right wing length between the areas, with area E being the highest, followed by area A, and then all of the others being kind of similar. And we could confirm that with a linear model, right? So we looked at the linear model and the overall p-value told us that that area was important, and then we could read off from the estimate column to see how the different groups ordered. So the overall p-value, small, suggests that the area has a difference, and then we can read off that area E is the biggest with 4 units higher than area A, which is the baseline, and area F is the smallest. And the other areas B through D are all fairly similar because they have similar coefficients. And we can visualize that with Visreg, right? Which is kind of an easy way to get the same information that you do from the summary table in terms of what the estimates are, and in terms of what their standard errors are, but allows you to visually see the differences really quickly. Okay, so the question now is, what do we do when we have more than one factor, right? So here we just had the one factor variable area. What do we do when we have more than one? So what happens is we do essentially the same thing. What we do is we put a block of indicator variables in for the first factor and another block of indicator variables in for the second factor. So we encode those categorical variables using the numeric indicator 0, 1 variables. So the baseline, the intercept in the linear model represents the baseline group for both, for all of the factors in the model. Okay, and anything else is then treated as a difference compared to that baseline. Now the overall p-value, the one down the bottom here, if we had more than one variable in here, so here we only have area, so we can use it, but if we had more than one, then that overall p-value is just testing whether anything in your model is important, and it's not testing the specific variables in your model. So we need something else to be able to assess the specific variables in the model. We can't use these p-values because there'll be more than one for each block, for each factor, right? For each variable. Right here we have 5. So how do we assess, how do we combine those 5 variables together? So we need another method of generating our p-values, and we do that with what's called the analysis of variance table, which we can generate using the anova function. So here's an example. We're going to put sex and area into our model. And this will allow us to assess whether area makes a difference after accounting for sex. So you remember that when we looked at the Petrels earlier, we found that the area E birds were largest. But in area E there were also more male birds. So it might have just been that area E birds looked larger simply because we were measuring more males and males were bigger, right? So we want to be able to assess whether there's an area effect after accounting for sex. So this is the type of model that we want to be able to use. When we look at the summary table, we've got a row for sex and a row for rows for area, 5 rows for area. So we can see that the baseline here, this intercept term, the 388.15, that will represent females in area A. Right, the baseline for sex and the baseline for area. Then each of the other terms are offsets from that, so differences from that. So the males then are 2 units bigger, birds in area F are almost 9 units smaller, birds in area E are almost 4 units bigger. Now the overall p-value here is measuring whether sex or area or a combination of them are important, but we don't have anything specifically measuring just sex and just area. Well, we sort of do here, right? Because sex only has 2 groups, we can assess its importance using the summary table here, because there's only the one p-value. But we don't have anything for area, although at least this is hinting as though area is highly important because all of the p-values we're given are highly important. So probably area will be important too, right? So the more formal way of doing this is to use the ANOVA function. So ANOVA is short for analysis of variance. And what that does is it gives you a table with just one row per variable, regardless of the variable type and regardless of how many levels are, or how many groups are within each of those variables. So here, sex is only a 2-group variable, but we've got a single p-value. Area is a 6-group variable, we've got a single p-value. So we can assess each of the variables in our model using a single p-value. So we can conclude that sex is important and area is important after adjusting for sex. So the process of assessing a linear model, assessing which variables are important when we have both numeric variables and grouping variables, but particularly when we have grouping variables, is you fit your linear model first. So that's the LM function. Then you do an ANOVA to check whether the things are important. And then if you've got importance, you can look at the summary table or use Visreg in order to determine how the levels differ. Okay. So the reason we do it this way is the summary table contains multiple p-values for the factor, so we can't assess overall importance. Now, one slight issue with this is that order is important, right? So in the summary table, and fitting the linear model in general, order is not important, right? Because we assume the equation for the linear model is just, it's additive, it's just a bunch of terms added together, and the order in which we do addition doesn't matter, right? 3 plus 5 is the same as 5 plus 3. Yeah, the order of the two numbers are not important. So the order of the factors in our linear model equation shouldn't be important, but they do make a difference in the ANOVA table. Okay, so the reason is that in the ANOVA table, just due to the way it's constructed, each of the rows are only adjusted for the things above it, not the things below it. Okay. So here we have an ANOVA run on an LM where sex is first and then area is second, and we can see that sex has a particular p-value and area has a particular p-value, right? But if you reverse the order, we're going to get slightly different p-values for each of those. Now in this case, they're not that much different. They're both basically telling us the same thing, that both sex and area are important, but nonetheless they are different. Okay. In general, this isn't actually a problem. And when it is a problem, it tells you something. So the advice is normally put the things you know make a difference in first and the things that you're not sure about last, right? So for example, in lab 9, when we looked at the lambs data, then we knew even before we undertook the study that male lambs are going to be heavier than females and that singles are going to be heavier than twins, right? We know that before we even undertake the study. But what we're not sure about is whether sharing makes a difference, which is the reason for the study, or whether the feed treatment makes a difference, but we'd assume that the feed treatment probably would, right? So I would put them in the order of rank and sex first, because we know they make a difference. And then we want to assess whether the other things make a difference after adjusting for those. So we put them last. Okay? That's basically the procedure. Right, so in the ANOVA table, each row represents a single variable, regardless of if it's a categorical variable with lots of groups or if it's a numeric variable. Each row is then adjusted for those things that are above it but not below. So that's why the order is important. If you change the order and things change quite dramatically, it just means that some of your variables contain information that contributes to the other one, right? There's correlation present essentially. So it's kind of useful stuff to know. But generally you put the things that you know make a difference in first, which are also the things that you're not really interested in. They're the things that you're controlling for essentially, right? So the things that you know are going to make a difference to your outcome measure, but that you're not particularly interested in, i.e. the things you know before you undertook your study, that's the stuff you put in first. And the things that you're not so sure about, which is usually why you undertook the study, you're interested in finding out if they're important, those are the things you'd put in last, because you really only want to know, are they important after I've accounted for all the other stuff that I know makes a difference. Okay. Generally, if you find something being important in the ANOVA table, that's when you then go to the summary table or visualize the model in order to characterize why they're important. Okay. So back here, Here's our summary table for the Petrels. So remember our ANOVA said that both variables are important, so we go to the summary table in order to summarize that. We see that males are 2 kilos heavier, 2 millimeters larger than females after adjusting for area, and area E is larger than area A after adjusting for sex. Okay. And we can see that when we visualize the model too. So here's the sex difference, and here's the area differences. So our conclusion here is actually quite a bit stronger than the conclusion that we've been able to make previously. Previously, when we looked at these data, we couldn't really tell if the area effect was potentially due to the sex effect. But now that we've put both of these things in our model, we can conclude that even after adjusting for the sex effect, area E birds are still larger than area A birds and area B, C, D, and F birds. So there does seem to be a difference in the size of the bird that we find, even after adjusting for sex. So in summary, when we have a linear model, the first thing you should do is when you collect your data, I haven't written it here, but the first thing you should do when you have a data set is visualize it, right? Is to explore it graphically in order for you to potentially find any issues that you may have with it. Okay, you can check for outliers and things like that, check if anything looks odd. And you can also sort of get a feel for what type of relationships you might expect to get out of the linear model. Okay, you then construct your linear model, you put the things in it in the order in which it makes sense. So put the things that you know make a difference first and the things that you're not sure about last. Then compute the ANOVA table. So run the ANOVA function in order to see whether things are important or not, right? So your p-values for important should come from your ANOVA table. If you've only got numeric variables, then the summary table is fine, okay? Or factors with 2 groups, the summary table is fine. It's only really if you've got factors that you should really deal with the ANOVA table directly. Then once you know importance, so once you know which variables are important, which variables aren't, that's when you can then switch to the summary table in order to find out how they're important, right? So, what are the estimated coefficients for those things? So, how much bigger are area E birds compared to area A birds? And you can use Visreg to visualize the effects if you just want to know kind of relative amounts. So the summary table will give you the exact differences. So, area E birds were 3.76 millimeters bigger. The Visreg showed us that they were bigger, but it was harder to determine exactly how much. I mean, you could see that it's about 4 units, isn't it? So you can get sort of fairly close, but you can't get the exact measures. And lastly, just remember that not significant does not mean not important. Okay? It may just mean that, that there's a lot of variation in your data, and even after accounting for the things that are in your data, you haven't accounted for enough such that that thing comes out as not being important. And vice versa, right? Significant does not mean important either, right? The statistical significance of something is really one of the least important things as far as science goes. It's basically saying that from my data I can infer something about the population, but the size of it matters, right? And the size of it does not generally come into the p-value directly. Right, the p-value is a combination of size and uncertainty, all sort of mushed into each other, and isn't quite the thing you want. What you really want is how big the effect is, and so that's sort of the more important thing to come out of a linear model is how big are the effect sizes. Okay, that's it.